I just wanted to open with just one question. Since we talked so much about the normal prion protein, PRPC, obviously we need that for prion disease because you need that to make further PRPSC, but is there another reason why blocking PRPC or downregulating PRPC may also be beneficial? Is there a toxic effect of PRPC?
Well, I know David Harris's lab has long said that you need the PRPC there to interact with PRPSC it's a subversion of the function of the PRPC that is involved in the toxic effect. So I guess one possibility is if you reduce the PRPC levels even later on in disease, you might reduce toxic effects by not having that PRPC there for its function to be subverted into a toxic effect. I don't know. It's just a guess.
So to play devil's advocate then, what if, we know that the PRPC, so you knock it out, not a lot seems to happen, but we also know they're more vulnerable to conditions of oxidative stress or other insults. And so if they have an ongoing production of PRP-scrapie, and is it possible that PRPC in the late stages is actually playing a protective role and we could make things worse? I think of Malucci's work in suppressing PRP after the onset of pathology and actually seeing improvement, which was fascinating, but doing it later didn't have that same effect. So again, I wonder about this point of no return. I don't know if we know. We'll wait for your data on your animals.
Do you have a comment on that, Roberto? Does anyone have any questions on the presentations?
So just following up on your question, Richard Axel's lab at Columbia University had quite a bit of press on a paper this past year where they showed that PRPC was integral in memory formation. Perhaps that might bear on your comments.
Yeah, maybe that's why I can't remember anything. Too many prions in the brain.
Can't remember that study?
I guess my first question is I have combined questions. Very, very interesting talks. Thank you for doing those. Byron, with your compound that can remove prions, based on the work from Stan's lab with David Peretz, where they showed that there was a big difference in infectivity between human prions and other strains of prions. You mentioned that you actually found bioassay reduction using the Charles Weissman's model where he inoculated the wires with prions. Did you use all of the same types of prions that you also found effective with RT-Quick with this compound?
No, not yet. And I mean, those are really long-term and expensive experiments, but I think one of the first tasks we have ahead of us is to do the in vivo bioassay experiments in the mice using the human strains especially, but also BSE and CWD to confirm that our RT-Quick results, the in vitro amplification assay are saying they're all knocked out, but I think for the important ones, before we really trust that, we'll need to do some bioassays in animals to address that question, that possibility.
Maybe, can I have the follow-up to that? So I guess related to that would be, what would be necessary to get something approved? I know when Stan was working on his agents to try for decontamination, I think in part he gave up because the bar was so high for being able to prove the absence of infectivity. And I don't know if Dr. Asher or others could comment on what would be required to get something approved that would be a decontaminant, because this certainly looks very promising.
Stand in a bucket of prions.
Where is David here? Why don't you comment on that directly rather than me jumble it all up?
Yeah, I'm David Asher from the U.S. Food and Drug Administration. I'm from the Office of Blood. and not from the Office of Devices and Radiological Health, which is the one responsible for hospital sterilizers, which is what we're talking about here. There are two kinds of, the FDA has authority for regulating the sales and labeling of products in interstate commerce, and we don't get beyond our authority. So we're very much concerned that anything sold in commerce be effective as described. And there are two kinds of claims that are entertained for sterilizers. One, a reduction in infectivity. And for those, one simply has to show, using some model system, that the log reduction factor is significant. For instance, half a log, that is, say, a threefold reduction in infectivity would not be considered particularly significant considering the variation in biological systems. But if one can drop infectivity by 1,000-fold or 10,000-fold, then it's more likely to have a reasonable effect in clinical practice. And the philosophy, and although I'm not in that particular center, the philosophy is that for a decontaminating agent, you can describe the results that you got, and you can market, although there is nothing at the moment marketed for spongiform encephalopathy with a claim for reducing the infectivity of spongiform encephalopathies, you can market with a description of what you got in experimental studies, providing a reasonable assurance, depending on the way the study was set up, that it would have a similar effect in clinical practice. The second kind of claim is more difficult to justify, and that is that the method produces an absolutely sterile product. And it would, we have no method that has, to my mind, justified such a claim yet, although obviously it would be ideal to have that, particularly for use with things like surgical instruments. As for the product that was described by Michael, it, as I recall, and it's probably more than 10 years ago, it did reach a panel review at the FDA, and there, as I recall, it demonstrated a significant reduction in infectivity but did not result in a sterile surface. The reasons why it was never marketed, you'd have to ask the UCSF and the institute that Stan Prusner founded that made the decision not to continue developing, and it was not an FDA rejection.
Thank you, David. Do we have any other questions about therapeutics?
So I have a question for Dr. Chiesa with regard to the mechanism of action of your compound. So it's known that copper and also other divalent ions bind to the octameric repeat region in the N-terminus of PRP, and that this also regulates initially endocytosis, basically. So do you think, or could you imagine that this is part of your mechanism? Because you have a zinc in your compound, right?
Yeah, it is possible. It is possible. It is something that we are trying to understand. So far, we have preliminary evidence that the zinc porphyrin that we are using binds to PRPC with 3 micromolar affinity. And we are trying to understand where it binds, if it binds to the octapeptide region. Or another possibility would be that it is becoming clear that zinc and copper can induce interaction between the N-terminus and the C-globular domain of PRP. So it is possible that our molecule interferes with this interaction between the two parts of the protein and somehow destabilizes the structure of the protein and makes it recognizable by a sort of quality control of plasma membrane protein, and that drives its rapid degradation That is one possibility. But we need to do basically NMR studies to understand where it binds and how it acts.
Just had another kind of broad question. In the field of Alzheimer's disease, we've learned that we have to treat people really, really early to get any kind of effect. And in fact, there are trials looking at people that don't even have any symptoms that are at increased risk. Given what you've learned from your studies and the importance of time, what is your prediction on how that's going to affect treatment for symptomatic patients versus people at risk?
Well, for one thing, we need really good diagnostic tests for all of those diseases so that we can know what's going on as early as possible, maybe even preclinically for people who are otherwise at risk. I mean, that's a real challenge, and my hope would be that amplification assays like the RT-QuIC is working for the prion diseases would be applicable in those cases, and then you would be able to tell patients who have or are at risk, even preclinically, or have early symptoms, which protein or proteins are misfolding, and thereby be able to get started with an applicable or a suitable therapy as soon as possible as those come along. I think that's incredibly important. I think that point was sort of made earlier.
And I would add to that, if we can know, what strain or strains are at play because of this possible specificity or shifting, and that you could, basically tailor a strain-specific type of regimen, I think, even potentially in Alzheimer's or these other diseases too, right? Where we might have a bit more time. And then the question too becomes about the genetic forms of this disease in particular. Can we predict what the strain will be just because it's genetic? I don't know.
Do you have anything to add, Roberto?
Mike's coming.
Oh.
My voice is really loud.
For posterity.
Okay, so it says the pharmaceutical stimulation of ADAM10. So I'm guessing that's a type of treatment, right? Because it says ADAM10 has recently been investigated as a treatment option in Alzheimer's disease. So it says here that they were using it as well for CJD. Well, the last time they did it was in 2014. Have they made any progression since then? My question is, if we've done trials, we've found certain medications that last for, let's say, a month or whatever, do we just give up, or do we continue to look at the same medication?
We don't
and
give
tweak
up.
it? What?
In reference to the ADAM10, that was actually a prior talk in the last
Yeah.
session. That's Herman. Maybe
Yeah.
he can talk about that. I can tell you, though, that in the field of prion disease, I feel like there's a lot more broader treatment targets than in things like Alzheimer's disease, which, to my opinion, keeps trying to do the same thing over and over again. So I do think there's more of a diversification of treatment targets in this field.
Go ahead.
Oh, I was going to say, on the same line, I mean, we talk about strains and shapes and the specific protein, but then too, and that's where I'm trying to more generalize, the actual, what's happening to the brain cells, and they're dying, and pathways for dying, and what we can try to add to that aren't the same conversion inhibitor over and over again, or a slightly different tweak on a conversion inhibitor, but coming more broadly. It doesn't mean those aren't useful, and we may need those as a vital part of it, but I think we have to think additionally on top of that, instead of just, the challenge is there's so many variables. If you, I can say, we'll just combine things. Well, you can't put 5,000 things into one brain and say what's working. So it's the challenge of doing it. But I think, that's where I think we have to go. So it's a change in focus, but we need individuals finding the tempting drugs and then start to rationally combine them. What were you going to say?
Yeah, I mean, along the same lines, I don't think we give up. You try to fine-tune, because fine-tuning can make a huge difference between something that's useful and not useful. And then doing the combination therapies, that's all of a sudden even when you don't expect it, sometimes you can see synergistic effects of drugs that are coming at it from different directions, even if they're not working so great on their own, as Val has been saying. So, and we have seen synergistic effects of two different antiprion compounds, aporphin and PPS, or was it a phthalocyanine? Because of what?
If it's a liability, like if you were to give someone that has CJD a medication and something wrong happens.
So the question is, if we give a patient with CJD a drug and it does something bad to them, that's the concern. Is that what you're saying? Is that your concern why we might?
You gave them a random medication.
Yeah.
You've done it in trials, it works on some, they, lasted 6 months longer, sorry. That's great, blah, blah, blah. And then, so it works for some, but then it doesn't work for others. So then we decide we're gonna rule it out and say, hey, we don't really know if it's gonna work for everyone. So we're not gonna approve it by the FDA. And most doctors will say, oh, that one wasn't working. It only worked for like 3 people. So we're not gonna, we're not gonna do that. So that's what I'm asking.
Sure, no, and I understand. And it's the challenge, and I was talking to someone earlier, it's the balance of science and medicine. And I am a scientist and a doctor, and some people here are too. And we try to be scientifically rigorous and get as close to certainty as is possible. But medicine is also an art, and we have to help people before all the science is in. And that's where it comes down to informed consent, and where you enter into a discussion. And I've had patients, we've talked about this too, Brian and I, about using bile acids in patients. Is there a role for that? Are they gonna work, are they not? We want to not do harm. We took an oath, right? Do no harm, but you also want to do good. And all you can do is present the patient, the family, here's what we know, here's what we don't know, here's possibly what could happen if you took this or used this medication, and go from there. And that's the best we can do. In terms of approvals and FDA approvals, that's a much more rigorous process, which, yeah, and that I can't speak to because I don't approve them, but it doesn't mean we aren't using medications or compounds outside of an FDA approved, but then you have to have informed consent for that. So, no, it's fine.
Yeah, thanks, Alana. I just want to remind people if they still have questions about any of the presentations, all the presenters will be here for the moving roundtable. So help me thank our presenters for excellent talks.